Hello everyone. Today I am talking about proteins, in which proteins having long chains that is present in a biological system. Proteins are very important in our day-to-day life. Proteins are divided into two classes, especially non-essential proteins, essential proteins. Non-essential proteins are those which are created by our own body, which are not essential because they created by our body, in which essential proteins are those which are created by another animal, plant, or all about that, in which we want them because we do not create or generate in our body. That's why essential proteins are very important to us. Now going to moving to another way, uh, types of how protein was made. Actually, proteins was made along polypeptide chain, in which one single amino acid is joined with another amino acid, and these joining are made by a peptide bond. These peptide bond having the C double bond NH group are combined called as a peptide bond. These are having at least 100 to 200 units, a single units. These all combined to call a proteins. Very small protein is insulin, which have 51 or units, in which proteins are classified into globular proteins, fibular proteins, and all about that. In globular protein, there is four types: primary proteins, secondary proteins, tertiary proteins, and quaternary proteins. Primary proteins are a simple polypeptide chains, in which they are very simple. They are linear in shape and all about that. Afterwards, a second one is secondary proteins. Secondary proteins is somewhat in which they are bent and just like a secondary structure was formed, a 2D structure was formed, in which a tertiary protein is a further enhancement of secondary proteins, and the quaternary proteins are fully complex proteins. Secondary proteins, we have helix models. These are simple and brief information about a protein and their classification. Lastly, come to a denaturation of proteins. Denaturations of proteins occur because of the change in the temperature, pH. Change in the pH means below acidic pH or basic pH can dissociate a hydrogen bond present in between the proteins. Let's create a primary structure. If I have a quaternary structure, if I have changed pH, the structure was distorted and it become a primary structure. That's why uh, change in the pH can make proteins shape dissociation. If a temperature was changed, if temperature was changed to normal temperature to high temperature, then in that case also uh, protein's shape was changed. That's why hydrogen bonds or breaks. These all happens because of the high temperature. For example, omelette. Omelette is a good example for when a normal temperature is there, a protein was in good shape. When temperature was high, the protein shape was gone. That's why a precipitation occur while in making an omelette. Thank you. Thank you for listening me. Thank you one and all.